Okay, hi guys. So sorry I'm not here today. I had to take Addie to a follow-up appointment that was important. So I'm going to record ahead the diagrams that you need to do today. And so you're going to watch this and complete them. So good thing is it's conceptually pretty easy, particularly if you did 1-2. This should be very much a revision of something you know from that. If not, it may have been a while, but it's just looking at the difference between prokaryotic and eukaryotic cells and the functions of organelles. So it is a lot in terms of we need to cover all of that in the one lesson, but it's not anything particularly difficult. It is SL and HL. So just a heads up, we are going to be drawing 3 cells. So we're basically going to draw the cells, and on each one we'll label everything, and then we'll also write the function of any things that are unique to that cell type. So we're not going to every single time we see ribosomes say makes proteins, like we're going to clarify that and then move on. So you may want, if you have some good colors, I do try to make organelles in different colors just because it's a little bit easier to see. But essentially we're just going to draw 3 different cells, label the things on them, and make a note of the function. And that's kind of what you need to know from this lesson. Okay, so we are going to start with a prokaryotic cell. So first up, we're going to look at a prokaryotic cell. Making sure my spacing's all right. Yep, that looks about right. So when we say prokaryotic cell, we're meaning either bacteria or archaea. We're going to talk more about bacteria. Archaea will come up a little bit more as we go into a little bit more ancestry stuff and some other topics as kind of a different group of unicellular prokaryotes that we think might have had kind of a role in the evolving of eukaryotes. So it's basically bacteria are the most important because that's what we have in contact with them, and also archaea. So those are going to be our 2 types of organisms. Okay, so we're going to start, I'm going to kind of draw the basic outline of it, and then we'll go from there. So these are much smaller, but I'm going to still draw it quite big just for it to be easier to you to see. But we may want to make a note that this is not to scale, and that these are actually very small. So not to scale compared to the other 2 cells. So these are going to be much smaller than eukaryotes. And in fact, when we get to drawing our eukaryotes, essentially the size of like 1 mitochondria in a eukaryote would be the size of this whole organism. So lots smaller than that. Okay, so then within that, I'm going to have a couple of layers. So, and then I'll end with 1 more. So I have deliberately drawn kind of 3 layers. If I was really smart with my colors, I would use the same ones on the plant animal as well. We'll see how I go, because I didn't plan quite that far in advance. So there's the 3 outer layers. So let's start with the innermost one is going to be our cell membrane. And just like in all organisms, it is going to be a phospholipid bilayer. So it would actually look like that. We're not going to draw it that way all the way across because that would be annoying. But we will make a note that it is a phospholipid bilayer. So membranes essentially look the same in all living organisms. And then we're going to make sure we know anytime we talk about a cell membrane, it's going to be what allows substances to both enter and leave a cell. And when we are talking about prokaryotic organisms, because they're unicellular organisms, this is particularly important for homeostasis. So I'm just going to make a note of that kind of point, that homeostasis, or maintaining that kind of internal environment that's consistent. And the cell membrane is important for all cells, but for a unicellular organism, that is the whole way that they maintain homeostasis, is by moving things in and out. So particularly important. Next thing I want to label is the cell wall. And we do want to know a little bit more detail about that cell wall. So that cell wall can vary, but if it's in true bacteria, it always has something called peptidoglycan in it. So pepti, like a peptide bond, and glycan, if you think glycogen, things like that, comes from carbohydrate. So it's a protein carbohydrate polymer. And it is found in all true bacteria. Archaea do not have peptidoglycan. They tend to have a unique cell wall that's kind of better suited for their environment. So in some cases, there'll be like really thick layers of peptidoglycan on the outside. So here's my really, really thick peptidoglycan, and it's sitting on top of the cell membrane. So then here will be my cell membrane below. So bacteria that look like that, we're going to call gram-positive. So this is a gram-positive bacteria because it has that thick outer layer of peptidoglycan. Not all bacteria are gram-positive. The other option is that they actually have this interesting adaptation where they actually have two membranes. So they actually have an additional outer membrane. Then they have an inner membrane. And then in between those two membranes, they have a thin layer of peptidoglycan. So they still have peptidoglycan, but it is different. So we call these, let's see if I can fit it. I'm gonna write this one above it, gram-negative. That's actually really important for how we treat them with antibiotics. So generally speaking, gram-negative bacteria are harder to treat because a lot of antibiotics work by basically finding the peptidoglycan and using to like break that down. So a lot of kind of normal oral antibiotics target peptidoglycan, and a lot of times they cannot find that peptidoglycan because it's below a membrane. So these tend to have to be treated with different types of antibiotics. So it's a really important medical distinction more than anything else. Remember, pepti, we said, means protein. Glycan means carb. So it's just a carb plus protein kind of polymer. So that's what the cell wall looks like in bacteria. Again, if it's an archaea who's still prokaryotic, which is like an ancient bacteria that are found in really extreme environments, they actually do not have peptidoglycan, but most of the time we are talking about these kind of more typical bacteria that we come into contact with, which would have it. And then the other thing it has on the outside of this can have is a capsule. And so the capsule is optional. So some of the ones that they may not have, I'm going to put like in brackets. I'll try and use like a distinguisher. So if I put it in a box, it definitely has it. Must have a cell membrane, must have a cell wall, can have a capsule. So it's like another outer layer. So it's a carbohydrate outer layer. So it's a carbohydrate-based outer layer. And essentially what it does is it helps it adhere to places. So it's almost kind of like a sticky layer. So it's something that kind of helps it stick on to other surfaces. So adhering, which is just sticking. And these are particularly, like we see this more with pathogenic bacteria that are trying to stick like on an organism. They tend to be more likely to have a capsule. Okay, let's add a couple other kind of weird outside things. So we said here that they can have a capsule. Another thing that they can have that also helps with the kind of sticking out is these things called pili. And again, this is something that not all bacteria have, but lots do. So they're little hairs. They're bigger than cilia. So cilia are really tiny. So pili are a little bit bigger than that, but still smaller than like the flagella. So that's going to be pili. And I'm going to put that in my little brackets to indicate that it's optional. And it does two things. So these are going to be small hairs. And they also can help stick to things. So they also can kind of get an attachment somewhere. But the other thing that they do is they can actually play a role in creating a point of contact between two bacteria. So they can actually connect kind of via their pili, and they can actually exchange DNA that way. So they play a role in bacterial sexual reproduction. So they also facilitate some DNA exchange. And that's going to be helpful for sexual reproduction. So bacteria do not have to reproduce sexually. They can reproduce asexually simply by dividing, but sexual reproduction is always going to be an advantage because it's a way of increasing variation and diversity. So this is a tool that can kind of increase their ability to do that. Okay, then I'm going to draw a flagellum. There we go. It has a little tail. So here's my flagellum. And it is again, optional structure. Not all of them have it. So it is kind of a whip-like tail. Its job is going to be to help with movement. And that's another thing that is actually considered like a criteria for life. So I wrote kind of homeostasis there. And this is another kind of criteria for life, which is a bit interesting because, of course, plants themselves and fungi do not move, but they have a way of moving parts. So they move their pollen and things like that so they can disperse, which is a way of doing that. So the idea of this facilitates that need for having some ability to move location. So their flagella helps them with that. Okay, so those are our outside things. This inside area in all organisms is always going to be our cytoplasm. And so the cytoplasm is the internal space. In bacteria, a lot of actual metabolic processes happen in the cytoplasm because they don't have organelles. So instead of in a eukaryote where you can have this happening in the mitochondria and this can be happening in the endoplasmic reticulum and this can be happening in the chloroplast, they actually, without those, the cytoplasm has to do more. So it's kind of a one area. So it's an internal space and lots of chemical reactions and metabolism happen there. And that's another kind of criteria for life. So living things have to be able to undergo metabolic processes. So things like cellular respiration. So check. For life, must do. So having a way of doing that's important. They can also often use their cell membrane for that. So a lot of times some of the reactions actually happen kind of across the cell membrane. So kind of a couple different places where that can occur. Okay, so then now let's look at what's inside of it. So inside of it, we need some DNA. So there is one large circular loop of DNA like that. So that is going to be the DNA. And it's often said to be in a nucleoid region, which is a little bit confusing because we want to make sure that we recognize they do not have a nucleus. So it basically means like a central area, but it is not a nucleus itself. So the DNA is kind of free inside the cytoplasm. Sorry, every time I get a marker, I have to come straight across it. So it's free in the cytoplasm. So in other words, not in a nucleus. The other thing that we noticed is that it was continuous. So it was one circular loop. And I don't necessarily feel like I need to every time remind us the DNA is the instructions for proteins. Hopefully having already done A1, we're good on that. But I'm just going to put it down in case that we need to be really clear that DNA provides all the instructions needed. So all living things have to have instructions for protein because they do grow. And so as a part of kind of the growth process is building proteins. We'll come back to that in 1 second. I do want to draw one other thing while we're talking DNA, which is plasmids. So here's my plasmids. And plasmids are going to be in my little optional category. So they don't all have to have plasmids. So plasmids are extra DNA. So they're extra circles of DNA. They actually cannot have any essential DNA. So all DNA that is necessary for life is found in the nucleoid chromosome. So in that one looped bit. But these might have like extra, I'm going to erase these pili. So they might contain additional adaptive DNA. And the best example of that is antibiotic resistance. So a lot of times the gene for antibiotic resistance is actually on the plasmid. And then that actually is helpful because bacteria can share DNA, but they can also really easily transfer their plasma to other DNAs. So they can copy the plasmid, so it's self-replicating. So they can take it and go, whoop, there's another plasmid. And then they can give it to another bacteria. Say, how's an antibiotic resistance? So that's part of why antibiotic resistance is really difficult to manage, because on top of the kind of natural selection piece is this plasmid piece where it can be transferred between bacteria quite easily. So those are plasmids. Lots of interesting implications for that with gene technology and so forth that we'll talk about next year. For right now, we just want to know what they are. And then the last thing that we need to add to our diagrams is something that all living things have, which are ribosomes. So we're going to add our ribosomes. So here's my ribosomes all throughout. And so my ribosomes are going to make the proteins. These are essential. So for bacteria to be considered living, they have to be able to produce their own proteins. So these are going to make proteins. And making proteins is a really important component of living things. So in order for them to be able to do anything, that's going to play a role in kind of growth. So be able to continue to grow and things like that. So a lot of these functions of living things, we can see this individual cell doing. So there's my prokaryote. I just want to particularly point out kind of the 4 things that it has that all living things have, just so we're really clear on that as we move into our next one. So for instance, I'll use red again, I think. All living things have a cell membrane. So that's what the all means. So I would not put that for cell wall because animals do not have a cell wall. Other eukaryotes don't have a capsule. So I'm just going to say cell membrane, all. Then all are going to have DNA. So not necessarily the nucleoid region, because in eukaryotes it's in a true nucleus, but all cells have DNA. All cells have ribosomes. And then all cells have cytoplasm. Cytoplasm has slightly different jobs, like actually does a bit more for a prokaryote, but general idea is it's found in all living things. Okay, so that's our first simple cell. So we're going to go from that and then do an animal and a plant cell. We're going to have quite a lot to label on the animal cell. Plant cell, we're only going to worry about adding function if it's something that's not in the animal cell. So we'll kind of cover everything pretty clearly on the animal and then just show with the plant what is distinct. So I'm going to erase this and keep rolling. Okay. Also, plants and animals are not the only type of eukaryotes. So we will actually, next class, talk a little bit, just pick up about fungi and just clarify ways in which they're like an animal cell, ways in which they're like a plant cell, and kind of chat about that. And then we'll make a mention of protists, which are actually so diverse that there's not really a lot to talk about them with. So we'll kind of at least mention the other 2 types. But of course, plant and animal are the largest of the categories, so that's why we're kind of focusing on them. So, animal cell. The other thing that we're going to talk more about a little bit more next time is the idea of kind of specialized cells. So when I draw this like one circular cell, animal cells are not all circular, so neurons have a long shape, skeletal muscles have a stretched out shape, box-shaped for epithelial cells. So depending on the cell type, it's actually going to have a unique shape. This is just our representation. We're just trying to represent that it's not as rigid as a plant cell, so it's a little bit more loose and flexible. So that's why we draw it as a circle. And again, not to scale because this is much, much larger than the bacteria. So this is a much larger cell. Oh, I should have drawn it over a little bit. Sorry, hold on. I'm just going to, so that I have enough space, I'm going to try to center it a little bit better on my camera. So again, we're drawing it to kind of just consume our space, but in reality, it is actually much, much bigger. So like we said, one mitochondria would represent all of that. And I don't know why I didn't draw it as a true circle, just to try to show it's not quite that concise. But there we go. There's my circular animal cell. So what I've drawn here, of course, is the cell membrane. And again, we're not going to draw the whole thing, because that would be all less than just drawing phospholipids. But we will remind ourselves that that whole cell membrane is in fact a plasma membrane. So when it is the outer membrane, we refer to it as the cell membrane. Plasma membrane refers more to its fluidity. So we can actually refer to like vesicles as having plasma membrane around them. So when we refer to the outermost thing, we will call it the cell membrane. And again, as we talked about, it is going to play a role in transport of substances in and out. So we already mentioned that, which therefore plays a role in homeostasis. The other thing that it's going to do more of in our animal cells is also going to do more role in cell communication. So it's going to enable multiple cells to communicate with each other. That's not really an issue in a unicellular organism because they only have one cell, but it is worth covering that that's going to be something that is distinctive about these. Okay, so we just have that one outer layer, so I'm not going to have worry about having my multiple layers out, cell walls, capsules, any of that. I'm going to go ahead and draw my central organ just to have it kind of placed. So that's going to be my nucleus. Okay, so there's my nucleus. Inside of it, I'm going to have a nuclear membrane. And then I'm going to have some DNA. And some of them I'm going to draw as little tight chromosomes, and some I'm going to draw a little bit more relaxed. So the more relaxed is going to be chromatin, which is not as tightly wound. And then the more kind of condensed one is going to be called chromosome. We're going to do a lot more on that as we do more cell cycle stuff, but just wanting to kind of clarify it can exist in one of two forms. So this is my nucleus. The nucleus has its own nuclear envelope, which is just its own membrane. That nuclear envelope has pores in it, so I've deliberately drawn it that way. Those pores are really important because that is how mRNA is going to leave. So when we're looking at protein synthesis, we have to have a way to make mRNA in the nucleus, but then get it to the ribosome. So the pores are really crucial for that. They're actually too small for DNA to fit through, but just the right size for mRNA. So again, more on that in gene expression. They also have a nucleolus, and the nucleolus makes ribosomes. It actually makes like the subunits. And then they kind of assemble outside because they would be too big otherwise to fit through the actual pores. And then, of course, it contains DNA. And in eukaryote organisms, those are linear DNA strands. And that linear DNA, I can't spell linear, there we go, is wrapped around histones, which we talked about in our DNA unit. So that should be a reminder. And then that DNA just basically depends on whether or not it's kind of in the process of being moved. If so, it will be wrapped tightly into a chromosome. If it's just kind of being used, it will tend to remain kind of unraveled more as chromatin. So we'll kind of draw a little bit of both, but that would be at different times. Okay, next thing let's go ahead and get on here is the cytoplasm. So this whole area is going to be my cytoplasm. Slight distinction, then we talk cytoplasm. The actual kind of fluid is called the cytosol. The area is the cytoplasm. A lot of times they get used interchangeably, so I wouldn't worry too much about it. So it's going to be filled with cytosol fluid. and that is just essentially going to be where all the organelles kind of move through. So they're all kind of submerged in that. And we do also have some chemical reactions. So really just glycolysis is going to be the main thing. We'll get to that down the road, but not as much happens in the cytoplasm here. Going with the cytoplasm, they also have a cytoskeleton. And a cytoskeleton is kind of like these fibers and tubes that kind of expand out and help give it shape. So they basically kind of maintain, so it's not too wobbly. They kind of hold it out, which prevents organelles from kind of collapsing in on each other. So it's kind of like spreading out little toothpicks throughout it just to kind of hold its shape. So that's going to be our cytoskeleton. So it's within the cytoplasm, but we can kind of count it as its own organelle. So it's going to be a series. Let me see exactly what I wanted to write about this. We're just going to do mainly that it maintains its shape. So it's going to be like filaments and microtubules. So it's just a series of little fibers and tubes. That's what I was trying to draw there. That are going to play a role in maintaining cell shape. They also sometimes anchor some organelles, so sometimes actually hold some organelles in place, again, to kind of prevent them from falling down. In some cases, they can help organelles move, so they kind of help in kind of the maintaining the placement of organelles. So we'll add organelle placement. Okay, so that's our cyto stuff. Let's go back up to the nucleus for a bit. So in the nucleus, we have our DNA. It is the instructions for how to make proteins. So that DNA is going to actually slip through as mRNA. It's going to leave the nucleus as mRNA, and then it needs to go to a relatively close by ribosome to be made. So we don't want my mRNA having to travel long distances. The cytoplasm is not a super safe place because of lysosomes, which we'll get to in a minute. But because of that, we would like it to try to not have to travel very far. So we want to make sure we have some good rough ER kind of near my nucleus. And there's going to be quite a bit of that, so it's usually covering quite a range. So let's draw some ribosomes to make it rough ER. When you draw the rough ER, we try to draw it more connected. And you'll remember that like we are drawing this, but we are recognizing our own limitations. So I did put in the back of your booklet a little key, partly because it shows you like a way of drawing it. And they actually didn't have theirs as connected as I would generally draw it. But it also gives you what it would look like on like a really high quality electron micrograph. So I have it so you can kind of see both. So you can be like, oh, this is how we would draw the nucleus. And we would make sure that we drew the membrane, the pores, and some chromatin in there. And then this is what it would actually look like if we were seeing it on a microscope image. So you have that as a resource. So this is kind of our version of this, so we're trying to kind of put it all together. So the problem with these is they're a little bit all separate, so we want to show what it looks like in one place. But you do have a resource if you want to look at like how do you draw different organelles. You have that in the back of your booklet. Okay, so sorry, back to where I was. So here's my little ribosomes. So on my rough ER, it should have ribosomes. There's also free ribosomes over here. And they're really tiny. They don't have a membrane. They have two little subunits. By the time we get done with translation, you're going to know that so well. So I don't need to worry about it terribly much now. So let's go ahead and label this as my rough endoplasmic reticulum. You're going to probably normally think of it as ER, but let's make sure we write it out a couple of times just so we know how to spell it for an exam. And then on my rough endoplasmic reticulum are ribosomes. So there's free or attached. So of course those are free, and that is attached to the rough ER. And we already clarified that ribosomes make protein. So I'm just going to write, just to phrase it differently, that they are the site of translation, which is the second part of protein synthesis. That's not going to be as meaningful now as it is at the end of the year once we've done protein synthesis, but then when you're revising, that will be helpful. So then my mRNA is going to pop straight out, boop, and it's going to land over here on a ribosome, and the ribosome is going to make the polypeptide. Then that polypeptide is going to travel through the endoplasmic reticulum to kind of get where it needs to go in the cell. So the endoplasmic reticulum, the rough one, is going to transport polypeptides within the cell. It doesn't actually have the ability to get them out of the cell, it just moves them around the cell itself. So it's like the highway of the cells. So I always use this analogy of a bake shop. So I'm running a bake shop. My bake shop is my ribosome. My recipes are my DNA. I've made my delicious baked treats, but now I need them to get out to people. So it would be helpful if my bake shop was located near a big road. So now my bake shop is right on the main road, so my Uber Eats driver can come, collect my delightful baked goods, and drive it along the road to where it needs to go. So the endoplasmic reticulum are that highway or that transport center. Sometimes some of the folding of the polypeptide actually happens there. So they will be made, the polypeptide, right on the ribosome, and then it will start to fold and so forth inside the ER. So that's kind of another interesting point. We also have smooth ER, which is going to look similar to rough ER, it just has no ribosomes. I am going to write ER for that one since we wrote it up above. The key distinction is no ribosomes. So therefore, it is not going to deal with proteins. It's going to instead be involved in lipids. So it actually makes, just like how the rough ER technically makes the proteins, it's the ribosomes doing it. So in this case, the whole smooth ER is going to make lipids. And that includes phospholipids, so that's going to be really important for like maintaining the cell membrane. So if the cell is like expanding or growing and you need new phospholipids, that's going to be the job of the smooth ER. It's also going to include steroids if it's a cell that makes hormones, so things like that, and then it's also going to transport them. It also has some random job in specialized cells. So in the liver, there's lots of smooth ER that helps with things like glucose regulation and detoxification. And there's also quite a lot of smooth ER in muscle cells that stores calcium. So it sometimes has some specialized jobs in individual cells, like in the human body or animals, but it kind of depends on the animal, because obviously not all animals even have a liver. So we'll stick with the general one here of making and transporting the lipids, but it does have a few places where as we get to body systems it might show up again. Okay, so there's all of that. If my actual protein needs to leave the cell, the ER cannot get it out of the cell. So again, I've made my beautiful baked goods. I drop them off at the Uber Eats driver and I'm like, hi, can you deliver these to my sister-in-law who lives in Canberra? And he's like, it's a long drive, but I guess I could. I'll just get in my car and keep driving forever and get to Canberra, deliver my baked goods. If I was like, hi, thanks so much, can you deliver these to my other sister-in-law who lives in Chicago? They'd be like, yeah, no. So what they would have to do instead is they could put it in their little car, but then they have to take it to the post office for me, ship it, package it, put her address on it, and send it off. That's the Golgi apparatus. So let's add a Golgi apparatus. So the Golgi apparatus is going to look like these flattened stacks. So it's going to look a little bit different. So over here, this side here, we call the cis facing, and it's going to actually receive the proteins or lipids. So you probably would have another one like down here. So I can draw another little Golgi. It should be kind of a little bit closer to the cell membrane, but anyway. So it's going to pick up the things. It's going to transfer it over here to the trans in, and then it's going to start making vesicles. And then those vesicles could leave the cell. So that is my Golgi apparatus. My Golgi apparatus is stacks of cisternae, which is just the fancy name for those little pancake stacks. and it's going to modify. That could be things just like putting an address, like tagging. So it's going to modify and package cell products, which is largely going to be proteins, but it could include lipids as well. And it's going to package them into a vesicle. So the little bubble coming off of it is a vesicle, and then that little bubble can exit through the cell membrane via exocytosis, which we'll get to when we cover cell membrane a little bit more. So we're going to package the products, put them in there, and then leave. All of that is going to take energy. So this process of running this cell is going to be a very energy demanding process. So we need to provide our cell with energy. So if I'm running my bake shop, I need electricity. So I've got my instructions, but if I don't have electricity, I'm not making any baked goods. So I need to have a power source. So all throughout, we need to have our mitochondria. So there would be lots of them depending on how active the cell is. So like muscle cells would have tons of mitochondria. I'll draw one in slightly more detail, like you can try to draw an internal space, or even just a zigzag inside can kind of show you that. So they're going to look approximately like that, and we're going to find them all throughout. I think they're more of a rod. I don't know why I tried to draw it specifically with that cute curvy shape. I just made that up. Sorry. Inaccurate. Bad. There we go. It's not quite as curved as that. Okay, so that is my mighty mitochondria. If you are sitting in an IB exam and it says describe the function of the mitochondria and you write the powerhouse, you will get 0 marks and you will make your examiner sad and mad. So we need to learn what that means. It is valid because it's referring to the idea of energy. So energy and power, that's kind of where it comes from, but we need to be a little bit more precise than that. So it performs aerobic cellular respiration, which is cellular respiration that uses oxygen. We're just making that distinction because when we actually get into detail in cell respiration, we will see that part of it actually happens in the cytoplasm. It has, again, this kind of shape. Inside there is an internal membrane that looks like that. And so then that area here is called the matrix. So it has like an internal area. Not going to go into tons of detail. It's kind of strangely placed throughout, so we'll have to learn a little bit more about it in B2 and then a lot more in C1 when we finish that. So in terms of the names for all the different sections, just for right now labeling that inside as the matrix. It has its own DNA and own ribosomes. We'll talk more about that next class because that gives rise to a theory on its origin. So there is my mitochondria. We're getting close to the end of this cell, so we need to add a few more things. So the next thing we need to add is some vacuoles. So vacuoles are just kind of small vesicles that store things. So they're going to store like water and food and also waste products. So they're just going to store kind of little good things inside of them. And they're often made by the Golgi. So that Golgi was like, here's a vesicle for you. And if you store things in it and start to collect things, then we call it a vacuole. So that is going to be our small vacuole. That's distinct from plants who are going to have one large central vacuole. So we've drawn a few small vacuoles. So we're going to store water. We're going to store like potential food in there. We're going to store waste and toxins kind of until they leave the cell. So they might kind of temporarily be stored in a vacuole on their way out. So then that vacuole could be expelled and that would be a way of getting rid of it. So it's just a little storage area for cells to use. Then we have a special type of vacuole called a lysosome. So it's also a vesicle, but I'm going to put X in it to suggest that it has dangerous enzymes in it. Ooh. So there's my lysosome. So again, it's just a vesicle like the vacuole is, but it has, instead of food and things like that, it has hydrolytic enzymes. So that's going to be my lysosome. And this is actually the first thing that I've drawn that is really only in animals. So we can put animals only. So plants generally do not have lysosomes. There are some examples where they do, but not most of the time. So it is basically a vesicle full of 40 plus hydrolytic enzymes. And so they are going to break things down. So they can break down organelles that are damaged. So if we have a mitochondria that's not doing its job, we're like, stop taking up space. And we just break it down. So they can break down organelles. They can break down intruders. So they're going to play a big role in the immune system when we get to that. So phagocytosis, so they'll release their contents, and that's how they can kill an invader who got into our cells. So they can break down organelles plus things that enter. I'm just going to write materials. So anything that enters a cell, in theory, those enzymes could break down. Sorry, ignore that. That's just lunch. Okay, another thing that is specific to animal cells, the plants, well, they have a similar version of it, but not quite the same. I am really running out of space. All right, I'm just going to make it work. We're going to give it some centrioles or centrosome. So with animals, it is specifically centrioles. Plants have like a centrosome region that can make microtubules, but not quite the same. And so I'm going to have to write about it all the way over here. Woo, sorry. Centrosome. And then in particular, ours contain 2 centrioles. And we will talk about those when we actually get to cell division. So they're going to release these like spindle fibers that help organize DNA during cell division. So for right now, I'm just going to put for cell division. So they play a role in kind of organizing our DNA during cell division. I'm trying to see if we have everything. That might be everything, which would be nice because that is certainly all the space I have. Hooray! That looks like that is a wrap. Okay, so that is all of my animal cell organelles. And then, like I said, the plant will be quite a bit faster because I'm not going to rewrite all the functions of the things that are the same. We'll draw them on, and we can label them if you want, but we don't need to reiterate their function because we pretty much have that here. Okay, so I'm going to erase that beautiful animal cell and draw as a plant cell. And if you want to use the same colors, that would be fantastic. I'm just not quite that organized because I'm going to try to remember what color I used, but we'll see how I go. Okay. Animal cell, no. Plant cell. Okay. Last cell. Okay. So this time I'm going to give it a boxier shape. That is true of most plant cells do have, because of their rigidity of the cell wall, they do tend to have a more geometric shape. It doesn't have to be a perfect rectangle. Some of them almost have like a hexagonal shape, but they do tend to be less round and a little bit more structured. So let's give it a cell membrane. I think we probably made that point, but I'm going to still draw a couple of phospholipids just so we do not forget that that is indeed a phospholipid bilayer. And again, I'm not going to give it a new definition. We've already talked about it enough. So cell membrane. I am, however, going to add my cell wall. So there is my cell wall. And my cell wall, we didn't even really clarify this on our prokaryote page, which I guess we should have. We'll do it here. What is the purpose of a cell wall? It provides protection. So it's a bit of protection. Kind of helps it have more structure and shape. So as we when we get to osmosis, we'll talk about the fact that like animal cells, for instance, can burst and plant cells can't because of having that cell wall. So it gives them a little bit more protection, which is really helpful. And then in plants in particular, the cell wall is made of cellulose, which we just did all kinds of fun time with. Cellulose is made of beta glucose. So we know all about cellulose. So it's a polysaccharide made of beta glucose that we talked about. We cannot digest it because we do not have cellulase. We have amylase, so we can digest starch, but we cannot digest cellulose. So that is what the plant cell wall is made up of. Okay, so before I draw the nucleus, I'm actually going to change it up for this one because for these, they actually have a really large vacuole that is actually often kind of pushes the nucleus even to the side. So they'll have one really large vacuole. And when you look at plant cells like in the microscope, sometimes it can even be hard to see the nucleus because if the vacuole is really full, it can basically be pushing it all the way out, kind of reducing its visibility. So that is going to be my large central vacuole. And it also can store other things. So it can also store water, food, waste, things like that. But it stores all of the things, particularly lots of water. And when it's full, it gives cells their turgidity, which is essentially just kind of their rigid shape. So it'd be like if you had a water balloon and you partially filled it, it'd still be a bit floppy. If you filled it as full as it could go, it would feel firmer. That's essentially what it's doing here. That's going to play a role when we get to osmosis and water transport, looking again at this concept of turgidity. So we'll come back to that, but we'll just kind of note now that kind of by filling up, it can give the cell extra firmness. That's what that means, and shape. Okay, then let's draw our nucleus. So here's my nucleus, and I'm drawing it with a nice nuclear pore. And it should be drawn basically the same as we did in the animal cell. So I'm just going to make a note that that is my nucleus. And again, I don't need to write what it does, but I will note with nuclear membrane and nucleolus, just so I remember all of those kind of subcomponents of it. Just like with the animal cell, near there, we are going to then want to have some ER. So I'm going to draw some rough ER. And we're just drawing these things again because we do want to make sure we know that plants have them. We don't want to only draw the special things and then forget that they have all of the other organelles as well. They can also have free ribosomes. And even though plants don't store body fat, because they have phospholipids, they will also have some, there's more rough. Generally the nucleus is almost covered in ER of some sort. They can also have smooth ER. So they're going to make their own phospholipids there. They don't use steroid hormones and they don't form adipose tissue, but they do need to create phospholipids. Sometimes when they create a protein that needs to go elsewhere, they will need to have a Golgi apparatus. So there's my Golgi apparatus. Ta-da. Okay. Let's talk energy. So they actually are going to have mitochondria, but before we draw mitochondria, so mitochondria cellular respiration is going to take food that we eat and convert it into ATP. For the plant though, it doesn't eat any food. So it is going to actually need to first make the food for the mitochondria to then break down. So let's make some food with some chloroplasts. And that's going to be my last of my organelles that are unique to the plant. Like so. And depending on the plant, they may have different numbers of them. So if it's a leaf, it's going to have tons of chloroplasts. If it's a stem, it might have some chloroplasts. If it's a root, it actually will not have any chloroplasts. So we do have kind of specialized cells. So how many chloroplasts it has will kind of depend on other factors. Ooh, I don't know what happened with that one. Goodness. All right. So there we have some good chloroplasts all around. So that is the chloroplast. I tried to draw it this way all throughout, but it actually has two outer, like an inner and an outer membrane, and then it has membrane stacks inside. So these little stacks are called thylakoid discs. And they are going to absorb sunlight for photosynthesis. And the purpose of photosynthesis is going to be to make the glucose. But glucose is not ATP. So we can't live on glucose alone, nor can plants. So they still have to have mitochondria as well. So that's just to get them to not have to eat. But then now they're also going to have the mitochondria. So all throughout, they're also going to have mitochondria because they still need to make ATP. Okay, so there's our mitochondria making ATP. One other little thing that we're going to add, and then otherwise we're done, is there is like a random kind of special organelle that you see in some parts of plants called an amyoplast that actually stores starch. So it's kind of like how we have adipocytes in human tissue, which are, remember, the fat cells that can store fat right there inside the cell. So plant cells have a version of that that stores starch. So it's an adipocyte. And so it actually stores, it's an organelle that stores starch granules inside the cell. So not all parts are going to have that, but it's going to be really important in roots and things like that. And sometimes they can be really large. So if we do look at like potato under the microscope, it's going to have tons of these. So it kind of varies by the part. But that's going to be a plant-only structure because animals, we do not store starch in our tissue or in our cells anywhere, but plants can. So that's another kind of specialized tissue. So just to clarify the four that are like plant-specific. So our cell wall is plant-specific. I'm just going to put a star NP. Or having a large central vacuole is plant-specific. So animals have them, but they're not large like that. Having chloroplasts is plant-specific. And then these adipocytes, oh, sorry, actually I wrote that word wrong. I'm so sorry, I'm glad I remembered. Adipocyte is a fat cell. So I wrote the animal, it's an amyoplast, sorry. So it's like a chloroplast. So it's an organelle within it. So that's where the plast comes from. And amylo, think amylose. So sorry about that. But we can clarify amylo is like amylose and amyopectin. And so that's a place where they can store some of that inside the cell itself. And again, depending on the cell type, that might be the whole cell basically. Okay, so I think I took pretty much your whole hour drawing those three things, but it is really important that you're good with these organelles as we start going into all of like the different processes, whether it's cellular respiration, photosynthesis, or individual kind of tissues and body systems. You want to make sure you're really clear on that. These questions, I'm going to actually advise you not to attempt them yet because we changed the order. There's quite a lot on magnification that we just haven't done magnification because we haven't done microscopes yet. So don't do the questions yet. Just leave them. We'll probably end up doing them as kind of revision at the end. And then next class we will go into this. It is like half SL, half HL, probably actually slightly more SL. We need to polish up some SL stuff. And then the HL part was pretty small, so I just chucked it in that lesson. So if you are SL student, go ahead and do all the way, and then you can stop in the last few slides. If you're HL, go ahead and do all of it. So, all right, thanks so much. Miss you guys, and I will see you tomorrow.